Hey everyone, what is happening? We are on to the action potential. This is how a nerve impulse it moves along a nerve. All right, so what we're going to do is we're going to start by drawing a little nerve so we have an idea what's going on. So what we have is we have our nerve there, and we have our dendrites, our cell body, we have our axon, and our axon terminal. Now, this axon terminal can lead to another neuron, or it could lead to an effector. This would be a muscle or gland. All right, and so we're going to focus on one part right here, and this section is called the axon hillock. So when we get some sort of sensory information, there is a change inside the neuron. There are ions inside the neuron, so these are charged particles, and they're also outside the neuron as well. And it's the movement of these ions that cause the impulse to be sent forward. All right, and so what we're going to do is we're going to break our sheet up into two spots here. And so we're going to have a neuron. So we've got our neuron, and this is going to be the axon hillock. All right, so that red spot. And so this first part, this is just going to be resting potential. And this is when no impulse is being sent. The cell membrane is negative, and the outside of the cell is positive relative to each other. All right, so this is where we find our ions. So on the outside, we've got sodium, and these are running all the way along our the outside of our neuron. And on the inside, we have potassium. The outside of the cell is positive, and the inside of the cell is negative. This is the resting potential, and this is maintained by sodium-potassium pumps. All right, so just over on the right-hand side here, I'm going to be drawing a little graph, all right? And this is going to just help explain the different voltages that are actually happening in our membrane. And so what we've got then is this is going to be the voltage over time. And so the resting potential of a neuron is at about negative 70 millivolts. And so this is resting potential. And so this is nothing is happening right now. No impulse is being sent. All right. So this is just you chilling. Your nerves are not doing anything at the moment. Now what happens is our next part is when some sort of stimulation happens. All right. And so this next part is called depolarization. And I'm just going to draw another neuron. It's the same one, but we're just going to draw it at a different stage. And so this is when we get enough stimulation to cause a change in voltage. Now, all our sodium is on the outside, and our potassium is still on the inside. During depolarization, the voltage-gated sodium channels, so when there's enough stimulation, this causes a voltage change, and this causes sodium to rush in through these voltage-gated sodium channels. And this causes a change in potential, change in voltage. So what happens is voltage goes up, 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 and it needs to get over this threshold of negative 55 millivolts. So as the sodium goes in, it's going to increase, increase, increase. And then once it gets to that negative 55 spot, it just goes whap, all the way up to about 70 millivolts. Okay, so right here is our threshold. Now, if there's not enough stimulation, then it's not going to get over this part, and this is when you wouldn't feel something, all right? So then once it gets past this negative 55, we get enough sodium in, then it causes our impulse to move along, and it's really a domino effect, all right? And so more and more sodium channels open because the other ones behind it also opened, and it moves down, it's propagated down the neuron. All right, and so this is called depolarization. So enough stimulation, like I said, it causes a change in voltage. This causes sodium ions to rush in via the voltage-gated sodium channels, and we get a huge change in voltage, and it goes up to about 70 millivolts, and this is when our sodium gates close. All right, and so then we're going to get into repolarization because the impulse has moved along. All right, and so we're going to go down to the next one. And so we have our sodium and potassium inside the cell at the moment. And so now this next section is called repolarization. And this is when potassium channels open up and potassium moves out of the cell into the cell membrane, and it drops the charge. And so what we've got is, I'm going to draw in some voltage-gated potassium channels. We'll just make those circles. And what happens is all our potassium moves out, and this changes the potential of the cell. And you can see that right over here. I'll just move up. And so what happens is we go down, down, down. And so this is our repolarization. And this is when potassium pumps are, or potassium voltage-gated potassium channels are open, and potassium rushes in or rushes out. Now, these voltage-gated potassium channels are a little slow to close, and so what happens is they don't just close right at negative 70 millivolts. They kind of overshoot just a little bit, okay? And so this is because they aren't closing as quickly. And so then we get this next section, which is our refractory period. And this is literally a reset. Okay, and I'll just step back for a second. The reason the voltage-gated potassium channels close a little slowly is this means that then the impulse can't go backwards. It's only one way. All right, and then the last part here is our refractory period, and we'll see that in a sec. This is when we have sodium and potassium pumps. This is a different type of membrane channel protein, and this is going to send all of our ions back to where they started. So now what we've got is the sodium-potassium pumps, and I'll put these in a different color. We'll make them triangular. And what these are doing is they are causing potassium to move back in from outside. And they're also causing sodium that had moved in to move out. Okay, and that gets us back to our negative 70 millivolts, and that allows us to be right here ready to go again. This is why nerves don't just fire, fire, fire all the time. It's they fire and then they have to recover and then they fire again. Now this is very fast, but they still have to recover. All right, so let's just recap here. Our resting potential was when there was sodium on the outside and potassium on the inside. No nerve impulse is being sent. All right, so the voltage, there's a potential difference between the outside and the inside, and that's negative. All right, so now during depolarization, some stimulation takes place, and enough stimulation takes place to get over that hump of the threshold, and then sodium just rushes in to the nerve. Okay, and this propagates that signal, and I'll just put this in yellow. It makes it move down the neuron just like dominoes, all right? And so sodium ions rush in via voltage-gated sodium channels, and then this more sodium causes this change in voltage potential continues to push other sodium channels to open, and it just rushes down the neuron. Now, to get back to where it was, we get potassium channels opening during repolarization, and potassium heads out of the cell, and it drops that voltage. And then it overshoots it just a little bit because those potassium channels don't close as quickly, and so it overshoots it, and then we get our refractory period, and then that refractory period is the time it takes for the sodium-potassium pumps, to get the neuron back to its resting potential. All right, so you can see that in this graph right here, and then the neuron is firing again. All right, so we will moving on to what it means to have a myelinated nerve impulse. And so what we've got, I just want to show you this. So with a myelinated nerve, we have a neuron like this, and myelin is covering over. And remember that myelin is like an insulation, all right? So we've got a myelinated nerve, and myelin is like insulation, so that's our myelin right there. And you'll notice that we have sodium on the outside still at our nodes of Ranvier, and there's some sodium channels at these sections, So when there's enough nerve impulse, it's the same thing. We also got to put in some potassium. So this would be at resting potential. And so when a nerve impulse comes along, we get a voltage change. And then what we get is we get sodium rushing in, and it changes the potential enough to get past that threshold. Now, what happens though is the myelin actually doesn't allow sodium to move in these sections. All right. And what happens is then it really it boosts that signal and it sends it right to the next node of Ranvier where we get sodium rushing in. And then it does the exact same thing and sodium rushes in. Now, at the exact same time as this impulse is being propagated, potassium is headed out through potassium voltage gated channels. And so it's resetting. And so potassium's moving out through its channels, and that nerve impulse is being boosted every single time that it hits a node of Ranvier. All right? And so then it's like skipping the line at Disneyland. You just boost right up there. Okay? And so then the sodium's rushing in, the potassium's rushing out, and then we go through that same refractory period where the sodium-potassium pump is resetting it, and it gets it back to its normal resting potential. All right, so remember that myelin is an insulation, and it really speeds up that nerve impulse. All right, please message me if you have any questions. That was a long one. I hope everyone is doing well, and we will see you soon.